Okay, so the last part on post-transcriptional regulation for this week, so don't worry because I'm basically dividing into 2 weeks. So for this first week, the end would be up until RNA editing. So next week, we're going to cover 3 more, the last 3. So for the RNA editing, it of course, as the name says, you could do, or basically the system edits. So it enables a cell to recode genomic information in a systematic and regulated manner. Apart from that, you could also selectively change the readout of a gene at single nucleotide positions within the primary RNA transcript. So this was first discovered through deletion and or insertion of bases. So of course it involves small guide RNAs, which are complementary to the target RNA in the mitochondria. And another type of RNA editing, which is called base substitution, which means that you substitute one-on-one. This is done with altered operating frames, so it would yield a protein with one or more changes in its amino acid. So there are 2 main mechanisms for RNA editing. First is insert or delete editing, or indel. Second is substitution. So for the insert and delete, nucleotides are either inserted or deleted in RNA, the name is obvious, and this is mediated by guide RNA. So if you remember back on the CRISPR technology, guide RNA is a similar thing. So this is done by base pairing with RNA to be edited, which serve as template for addition or removal of nucleotides, and this is where the idea of CRISPR comes from. Secondly, substitution editing. So this is done by chemical alteration of individual nucleotides. So it involves, of course, enzymes which recognize a specific target sequence of nucleotides which catalyze the alterations. Now, these are some of the important features of RNA editing. So not restricted to this, there are a whole lot more, but I'm just highlighting some of the important ones. So it could occur in mitochondria and or chloroplast, that would be in the case of plant. RNA editing events occur at random transcript. So both 5' and 3' non-coding regions of mRNA may undergo editing. Apart from that, structural RNAs such as tRNAs and rRNAs are also affected by editing. So editing can convert a trip codon to an arginine codon from CGG to UGG, kind of like your agarboot. Okay, so UGG codes for tryptophan, but mostly in plant mitochondria. CGG codes for trip. Okay, so don't be confused because in plant, RNA editing is actually a bit mind-boggling. So don't worry, I'm not going to cover it. Again, RNA editing in plant is very complicated matter. So some other feature, start codon, your AUG, your methionine, can be created from THR. ACG is just a matter of replacing the C or substituting the C with U. Stop codons can be created by editing CAG, CAA, and CGA, whereby you replace the first C with U. Okay? So the most frequent amino acid substitutions are from proline to leucine, from serine to leucine, and from serine to phenylalanine. So plant mitochondria do not use the universal genetic code. That's why you can see this. Okay, so they have a code of their own. They do use the genetic code, the 64 genetic codes, but there is a variation to it. Now, these are different forms of RNA editing, okay, in organisms, the substrate and the mechanisms, as you can see. So taking an example of the insert deletion, so we will go to the substitution in a while. For insert and deletion, for example, okay, U and also D and U, okay, this is in chitoplastids, so in plant. So substrate is the mRNA. So the mechanism is by cleavage or ligation. So for the C, U, G, and A, this is an example of the organism. So substrate is actually in the mRNA, tRNA, and RNA, but mechanism is not really understood. So for the conversion or substitution, C2U, okay, usually this is in higher plants, okay, same organism, and in some mammals. So most of the time involve mRNA, sometimes tRNA or rRNA. So a lot of times in plant it is caused by deamination. And the same goes in mammals, but in this organism it is actually not known. From U to C, okay, this is usually in mammals, okay, and mRNA, but mechanism is unknown. In higher plants, mRNA as well, but mechanism is unknown. So from the A to I, this is only in mammals. Well, I would say unique to mammals. substrate is mRNA, and this is done by deamination. Now, for those of you who wonder what is I is inosine. How the heck do we have an I whereby we only have A, C, U, and G, right? Okay, so think about that. Now, in mammals, RNA editing is mainly represented by C to U and adenosine to inosine. So this is basically I, okay, functions as guanosine. Conversions, okay, of these conversions would generate single amino acid changes in the resultant protein with significant consequences for protein function. Whether it would make it function better, more efficient, or probably not so much better. So among mRNAs that were found to undergo editing, the best characterized examples are alipoprotein B, ApoB transcripts, whereby the substitution is C to U, and messages for neuronal glutamate and serotonin receptor subunits, okay, which is adenine to inosine substitution. So RNA editing is of course now recognized as an important mechanism for generating molecular diversity, and this has been made possible and utilized widely in CRISPR technology. So inosine in mRNAs was detected only after RNA editing. However, inosine at the wobble position of several trans RNAs in the main molecular determinant, okay, is the main molecular determinant for the degeneracy of the genetic code. So, all in all, RNA editing allows for the expression of several protein variants at the same time, but it would depend on the number and combination of modifications introduced into individual mRNAs. So this is actually a very interesting topic, okay, but complicated in a way. So I would leave you to that. So if you are interested, okay, in RNA editing, okay, so probably would have a discussion about it later on. What I have here is basically just a summary of it, because if I were to go too deep, then it will be very complicated, because it is complicated.